The FDA has awarded AnaBios a grant for completing the qualification of a novel biomarker that will enable regulators to assess the potential cardiotoxicity of new drugs in advance of clinical studies. This is very critical because it will improve over current methodologies, which are not very predictive, and will help ensure that only safe drugs will enter clinical studies. The biomarker is obtained by measuring the effect of new drugs on human isolated cardiac cells in the laboratory, and by measuring how the drug changes the physiology and the behavior of the human cells, it is possible to then predict whether or not the drugs could pose a toxicity risk to patients. Current strategies rely on the use of animal models for predicting the potential cardiotoxicity of new drugs. And this approach is not particularly predictive. So what happens is that sometimes toxic drugs make it through and progress into clinical studies, generating adverse events and risk for patients, or in other situations, potentially useful drugs are left behind because of false positive signals into the animal studies. So by utilizing human cells and human tissues, the novel biomarker will actually increase the success rate of clinical studies, ensuring that only the best drugs can progress into clinical development and ultimately can reach the market and larger patient populations. There are specific molecular and physiological features that are unique to the human heart that are not well replicated in animal models. And this becomes a problem when trying to test in a quantitative fashion the effect of the drug on the heart or the cardiovascular system in general. And so by utilizing tissues and cells of real human origins, we have a much higher probability of generating truly predictive data.